Okay, I forgot to record to the cloud. So today we are going to finish the lecture, which is immunoprofiling analysis and how the experiment procedure was conducted. And then we will download some T-cell receptor single-cell RNA-seq data to see the data type and how can we proceed. And then I switch gear and talking about the Julia language. And we will set up our running environment on our computers. And then we will use a Julia language-based software tool. This is for using mutual information to construct the gene regulatory network and see how this program can be used. Okay, so we finished last lecture in Tuesday. It's about introduce adaptive immune system. This apply to human and the mouse. They have similar structure in those immune cell receptor gene reservoir. So for T cell, for example, you have different subdomains, subunit, and then the subunit, you have a different variants of this gene. And for different pieces. So that gave you the foundation to create the huge reservoir of the diversity. So you can have up to 10 to the 21 different possibility combinations. So this space allow you to have your T cell in regarding to their surface receptor subtypes. So every T cell may be considered a unique combination of one of this. However, once you have been affected by certain pathogen, then this landscape of diversity will shaped by the pathogen, and the particular isoform of this T cell receptor content T cells will get expanded, so the population. So you will see a lot of this expansion of colony of certain types of T cell with specific T cell receptor combinations. So when you have information about the individual cell and what their T cell receptor subtype looks like, you will be able to compare the frequency of different sub isoforms of T cells and have some idea what T cell, what combinations of T cell receptors respond to the infection, to the pathogen. Okay, in order to do that, and 10x Genomics has this amplification kit released in 2018. So before that, it will be very difficult to have that information at a single cell level. So you may have pulled samples and then using deep sequence, you can get some of this rays, and you may want to classify this rays and group them and look at the frequency. So that's kind of a pooling strategy, because if you don't have a single cell level analysis, you have to do pooling, and you compare, for example, patient or before and after exposure, and you see the frequency change of certain types of the subunit of T cell receptor or B cell. Then this is kind of a pooling, and but now you can do single cell level and with this kit. So I'm going to show you. Most importantly, you do this in parallel with the single cell, the conventional single cell RNA-seq. So you have a transcriptome information for all the T cells, and also with their T cell surface receptor combinations information. So you have both. Okay, so this is the same idea, and I already showed on the surface of T cell or B cell, you have either alpha chain, beta chain, and the alpha chain, beta chain have different combinations of those subdomains of peptides. Similarly, in B cell, you have a heavy chain, light chain for the antibody. Okay, so for 10x genomic solution for this, the first step is the same. However, they use this, the difference about this bead is this maybe is a 5' kind of design. So it's not a poly T. Remember, we have previously said, okay, this bead was designed to fish out poly A-tailed mRNAs. Here they use this so-called complete switch oligo. Okay, the rest of them are the same. You have this bead preparation, and then you have partition oil, and you try to capture individual cells. In this case, we will focus on immunocells. Okay, you can have blood cell drawn, and here you capture either B cell, T cell, or with some other cells. But we will be able to differentiate the T cell out of other populations. And also, so that's the design for this template switching oligo. And so now that's the procedure. You have the same design for this 10x barcode to tell you which cell this is using the barcode. Then you have UMI that will tell you the unique molecule you captured. And then this 13 base pair template switch oligo is next. Then you have this specifically designed reagent to do reverse transcription. Okay, and their poly-T primers, which here I don't know exactly how this was designed. It looks pretty sophisticated, but anyway, at the end you join your switcher oligo with this, and you can start to amplify this piece of mRNA. It's actually already reverse transcript, so that's cDNA from poly A. So step one is largely the same as conventional setup for this 10x genomics CRIM. And the step two is to do again the same, and you have a clean up and QC, and by after this broken this droplets and pull them and start to do the reaction in the mixture and then recover. Okay, step three, start to do performance cDNA amplification. Okay, so at this stage, the droplet, the GEM has already been broken. So everything has been put together, so you can amplify this, and using many rounds of this amplification, and you get sufficient material. So that's amplified cDNA fragments. you get enough for this, for the sample to be separated for downstream, and you have the same amount of this, and then you will separate it into conventional single-cell RNA-seq transcriptome, and the T cell receptor. If you want to do B cell receptor, you can separate it into three portions. So you have enough this amplified cDNA, and for you to construct multiple libraries. Before, we only construct libraries for gene expression library, but now you can construct a library for T cell receptor and the B cell receptor. Okay, so that's the key. And for the same cell, you have multiple copies of their cDNA content, and then you can separate. Although they're still labeled to the same cell, maybe same gene, but you have multiple copies, so you can separate. Okay, so that's diagram illustration. So that's single cell droplet. GMs undergone reverse transcription, then generate 10x barcoded cDNA. Okay, now it's still in the intact droplet, and then we will yeah, so all the cDNAs in the same droplet contains the same 10x barcode, indicate they from the same cell. Now, the next step is to the partition in the emulsion are broken open up, and then you put them and then mix them freely. But because they still have a 10x barcode, you can still track down to which cell they come from. And then you amplified this material, get enough cDNA amplification, and it's ready to be split for separated analysis. So if you only look at single cell RNA-seq, the gene expression, normally you just go this direction, you put here to do the downstream. But now we are going to split half-half. One half into T cell or B cell receptor analysis, another half to the gene expression. Okay, so gene expression, in this case, we will get the matrix of gene, and they're in the cell expression level. And in this side, you will process it slightly different to amplify the T cell receptor gene and in another round. But because for the same cell, you still have the same barcode here and the same barcode here, later you can link there. That's the same cells, T cell receptor isoform with the same cells expression is supposed to be the transcriptome level. Okay, so this cartoon is important, the most important part. So I split the amplified the solution into half here, half here. Okay. And to process them downstream separately, independently. At your left, it's called VDJ library preparation. Okay, because we are looking the sequence in the T cell, B cell receptor genes, and we want to find their VDJ isoforms, the combinations of those. Well, on the right side is still gene expression library. Okay, that's we already know a lot of this. Okay. So next step is important. I will leave you a couple minutes to think about this. So for the tube, on your left, we are going to amplify cDNA use PCR. But this premise of PCR was specifically designed only amplify the T cell receptor region. Okay, on your right side was for the whole transcriptome, but this side is only amplified T cell receptor or B cell receptor, depends on which type of library you are preparing and want to analyze for. So take a look at this and think about it. Any questions so far? I hope you know what we are trying to do here. So in the blood cell, you may have mixture of T cell, B cell, and I think their treatment for both of them can be amplified. Okay, the receptor. Then they have this fragmentation and size selection to enrich this production of this TCR segments amplified. Okay. So the final library contains this Illumina sequence standard primers. So you can next is to send this prepared library for Illumina sequencer to get the massive and the large scale sequence. So you can have enough reads to do the analysis. Okay, on your right side is conventional. Remember, we have two tubes left for the VDJ library, and on your right side is still the transcriptome, the gene expression library. So that's the same as what we know before. Okay, so once you have these two tubes and prepared, you have two libraries for the same sample. And you will send this prepared library. One is called single cell VDJ enriched library. That's for the receptors. And this is a gene expression library. So you send this to Illumina sequence to get back all the reads, and you can still track down the same cell. So because you have text barcode, okay, you prepared from the same population of cell, and not the same population of cell, but it's exactly the same cell because you labeled it, but they only separated their amplified products, right? Okay, so once you have your sample sequence information back, and now we are going to do VDJ T cell, B cell analysis. So Cell Ranger is still the one that can help you, but that's just another module. And normally you run this, make FASTQ, so process this into the FASTQ cells files, and then you specify this VDJ as a parameters, and then do this I would say it's still kind of a mapping procedure, but specifically for VDJ receptor genes. And then yeah, so you still need to have reference to tell, to give those gene and isoforms information to give this, so that the alignment or mapping can be done. Yeah. Okay, so when you run this, you will see a lot of output files. Okay, you have summary of this. This is similar to gene expression, single cell RNA-seq. And then you see this clonal type. Okay, they all sequence of different combinations of T cell receptor genes, VDJ combinations. You will see them. And that's mapping, and they tell you where this row sequence was mapped to. The most important file, I believe, is this consensus and annotation. Okay, so a lot of them are the intermediate level of files, which from the pipeline, the one step to another, mapping and assign receptor genes, subtypes, and at the end, you will have this. Let's look at what's inside this CSV files. So that's this file. So all contig annotation SV CSV. First, you have this barcode that indicate the cell. Okay, so you will use this barcode to link to your transcriptome gene expression level cell. But in this file, it's only tell you what the T cell receptor gene, so this alpha unit, and that's its alpha unit. Let me see. So that's V, no D, so it's V and the J. That's V3 and the J27, and then the alpha constant. So that's this combination, and then this is a yeah. You can see this specific CDR sequence, which is the marker of this type. And then you will be able to get UMIs. Yeah, so how many? Basically, that will be the frequency, I believe. Yeah, so every single cell here is linked with so-called contig. Contig is your assembled T cell receptor combinations of those sequence. And then you have the VDJ combinations, the type. If you really want to look at what's inside, you can track down and to the full length sequence of clonal type 1, for example. You can look at the sequence. Indeed, you found this particular consensus, and you know it gave the subtype of this. So for this particular clonal type, you also have its beta unit look like this, so you know its VDJ combinations, what type. So once you have this information, because you have all the cells, and then the question you can ask is, if I want to look at clonal type 2 associated cells, so this clonal type 2, we have alpha, the beta chain. And we say, okay, which T cell in my population belong to clonal type 2? So I can just check this table and found the cell which in this group and highlighted. So in this case, you can see majority of the T cell are highlighted by this clonal type 2, because that's one of the most frequent clonal type. So that tells you that this particular response of the T cell in this sample is specifically and amplified this clonal type 2 T cells. And if you know what pathogen and has been used to challenge and get the sample from, and you will have idea about why, idea about this landscape, the change between the frequency. Okay. Then you may want to go further and to look at this alpha chain and the beta chain subunits and see, okay, why this particular type get amplified extremely high frequency to this extreme high frequency. Okay. So that's a typical clonal type frequency you will get. You see this the majority one appeared in most of the cells. And this immediately you have a quick drop about the frequency of particular clonal type within the population of cell. And then you can present in this way. So this is the most frequent one, and this same clonal type in most of the cells. And then the second level, which is this one here. So that's the second, that's first, and this maybe third. So you see this relative number and to show the proportion. So that is the generated clonal type frequency landscape. Okay, that's all the finish up of the lecture. So I'm going to go to 10x Genomics website to download a set of example data which generated from this immunoprofiling analysis. So what I did, if you want to repeat what I did, you can Google. If you Google 10x Genomics dataset, then you should be able to be directed to 10x Genomics resource datasets. You can also get datasets from publications in the GEO. You should be able to find it, but here is easier for me. If you want to download, you may have to do a quick registration to tell them your institution, your zip code, something like that. I did before the lecture. So here you can see they separated their data set into by the products which used to generate those data. So that will be the single cell gene expression data, and the second one is immunoprofiling. So inside here, they have provide quite a lot of different data set. I think this will be the earliest and 5' gene expression VDJ, and so that's use V version 1 chemistry. They can also have a cell surface protein, so you can, you have sufficient amplified the solution and the mixture. You can separate it into gene expression. You can do T cell, you can do B cell. You can also work for the particular cell surface protein markers. And so for the same cell, as long as you have enough material you generated from single cell amplified, you can separate them into multiple downstream analysis. So so what I did, I registered and opened one of these. This 10K human DPMC sample, 5' version 2 chemistry, single cell profiling dataset. So it tells you where the sample comes from. It's a human periphery blood mononuclear cells, and from a healthy female donor, age between 25 to 30. Then they use gene expression and VDJ library, generate 2 libraries from 16,000 cells. And at the end, 9,391 cells were recovered. So the gene expression analysis was done with a 5' reagent kit. And the TCR amplified with a certain number of rates. And we also did B cell receptor for the same one. And for gene expression level, you have more rates to be in there because you try to capture the whole transcriptome, where the T cell, B cell, you only focus on the receptor genes. Okay, so then the view summary is, that will be the similar as your let's see. So first we have this gene expression. Number of cells, rate. Okay, so that's the gene expression panel. You have those statistics, you have t-SNE, library size, color coded, and then clustered into different clusters. Okay, most of single cell RNA-seq data will end here, but because this has been done using both expression and T cell receptor profiling, B cell receptor, so you have another tab here. In this case, so you have total 9,000 over 9,000 genes, and estimate T cells, there are 4,500 T cells here. And then not every T cells can successfully get this VDJ pairs, because you have to match the alpha and the beta unit. Some of them maybe just miss one of the units, so you cannot have a full picture. So here, almost 4,000 T cells has paired alpha and beta chain, so you have a full information, so you can analyze them. 87% of has both. Then you just count your CVS files, and you see, okay, the most frequent clonal type 1 has 27 cells, and the second one have 12, something like this. And also you have the marker sequence in there to indicate which alpha beta chain subtype they are. They're very similar. You have a B cell, and the number is small, it's only 436. You get information from that's the IgH, IgK sequence, antibody combinations from different immunoglobulin genes. But same idea, okay? So after sample, I click libraries, and then there some information about the sequencing, saturation, and the library for gene expression analysis is called the GEX normally, and for this is VDJ_T, and then library for VDJ_B. So you have 3 libraries constructed for the same sample. Experiment design. So can't see that clearly, but that's a separation. Yeah, you see this separation of one GM, can't see GM wells, and then separate into 3 libraries construction, and you generate different libraries. Yeah, I think that's about all I want to share with you. Do you have any questions? Online students? Brennan says, okay, yeah. So I think I can show you if you have the information about the clonal type. See, I have a label, CO1 is clonal type 1, CO2 is clonal type 2, CO3 is clonal type 1. If I have this vector label, how can I highlight it in MATLAB, the environment? So I will show you an example how you, if you have the data like that, what you can do. Just an example. Once you have this data, you want to show in your scatter plot and label different clonal type in different color, and see which color is more frequent and where they are located. So I'm going to use example data. So this data is from blood cell, and we do have a B cell, T cell in there. Okay, so for instance, if you have experiment and you measured this cell population expression level, so you know how to do this. You can show the gene expression level for different genes. I don't know if we have a T cell receptor genes, TCR. It's not here, so there's lowly expressed genes. So you know you can do this. So our job right now is how can you label the cells using color depends on their clonal type. So we need extra information to color them, right? These two provide, let me see, this labeling. So there's a way you can label. Let's see where. Yeah, so we should have an option that here you can select external variables to allow you to color the cells. So here you have different colors. You can cell type, you color them this way. If you select cluster ID, so you color them differently. So this depends on this variable here. So if you batch ID, there's no batch ID. So what you can do with this is we need to create external variable which contains the same numbers of indicators for each cell. So here we have 2,509 cells. So I need to create a variable, a new V. Say, okay, this new variable that sets all the zeros. I need to have 2,509 cells. So this new variable is here. Let me see if I can have that in there. So once in your workspace have this type of variable, and this program recognize there's something in there with the same numbers of the rows, number of cells. So this will show up. You'll be able to customize a new color code for C. So now you can select this variable to use this to give the color. Now, because I don't have any values for this new variable, it's all zeros, so that's the color. But if I say, okay, I want to put a new variable 1 to 50 as 1. So I put first 50 cell as 1. Now I can see, okay, I want to customize C and then select this. And now I would be able to see, okay, there in this variable, I indicate there 1 and 0. There are 50 ones. You can find there one there in some of this in color coded in the orange, right? So that's the way if you have an external variable vectors, you can read that in and to color code your cells. So what you need to do is to for for your cells, then you put zeros first as a background, then you start to add those clonal type for T cells. And then use other code for clonal type for B cells. In this sample, you have both T cell and B cells. Okay, I hope you get the idea. I use another color, and then I should have 3 colors. Okay. So I will leave 5 minutes for questions and feedback, and then we take a break, 5 minutes. You may also try random variables. So if you have maybe normal distributed. Okay, in this case, you have random variables, and this every cell indicated with different colors. Okay, 5 minutes break. Come back at 11 sharp. Okay, so we are going to finish off this MATLAB thing today and switch to Julia, a new language. So this is a new programming language, pretty new, maybe only 5 years old, and invented by some MIT professors. I would say this is a beautiful language, and a lot of things make much sense than R, or especially R. It's high performance. That means to deal with this large scale data easily, much easier than R, and the native language is compiled and then have high performance. So one of the fastest language you can have to deal with data. A lot of features good for programs as well. I mean, the population of user is much smaller, maybe 1/10 of other languages only, because it's new and but it's very active group of people. So I want to spend some time to share with you about this configuration and the setup of everything. I think I only mentioned this once, and then if later you decide to pick it up, you have the chance to take a look and have some quick idea of what it is. Okay, so if you are on this page, we are going to just download this. We are using Windows, so please click 46-bit installer. Then you can run this from your download folder by double-click it. Then just click and using the default setup, then you should be able to get it installed very quickly. So you have this new application called Julia, then you can just click it and have this window. That's all. And you installed it and ready to use. The language itself, similar as MATLAB, and may have some flavors of Python. So in this case, I'm going to use larger font size so you can see. Well, let's see. You can do this, right? Then you can see assign. Remember to put a comma here. So that's you you define a vector. So it's very clear you define it's vector or it's horizontal matrix with a one dimension, so row column. So the data type is really clear here. So you only mess up with, are you having a matrix with just one column, or it's a vector, column vector? Because you have the data type indicators here. There's 3-element vector with floating precision, floating point files, numbers, 46 bits. So if you see, okay, I have a B, which is only 1, 2, 3, then it's okay, there you only need integer, so that's integer, and you know this takes different amount of memory. So if your input data doesn't need to be floating precision and the high precision, then it will use just integer. That save much memory, right? So this language is more precise. The control, the memory, the process, and they give you this slightly lower level control for your data, right? Let's see if you do a C, if you don't put a comma in there, so that's a matrix. So you see, okay, the first one, because I've put the comma to separate my data, then I get a vector. And then in data science, in machine learning, we like vector because we always play with the data points. But here, if you don't do that, you get a matrix C, okay? I'm trying to do this vector multiplication element by element. So yeah, so you can do that. And so you remember you have A, which 1.2, 2.5, 3.5, and multiplicate by 1, 2, 3, and element by element. So you put a dot here. You can do that. So if you do, and you see the result will be a floating 64, this data type, instead of integer, because they're not integer. So I transpose, let me see if I can do this. I'm not so sure. So that's a dot product. Basically, you transpose your vector, make it a row vector, and then multiply it by another. So that will be the dot product between two vectors. I make a random matrix. Okay, this is a 4x4 matrix. Then I can transpose it and plus itself. Okay, I transpose the matrix, add it up, and then divide by 2. That make symmetric matrix D. So I want to use eigenfunction to get eigenvalue. Let's see. Yeah, so I get 2 errors. One is I use this MATLAB type of this syntax, but it looks like it's not allowed. And then see, it gave me eigenvalue and eigenvector eigenvalue, but the eigenfunction hasn't defined. So I miss one step, is using linear maybe I haven't installed. Yeah, so I have to install this linear algebra package. So what I can do is to type. Okay, so if you type this, what do you call this? Squared block bracket, squared bracket, and you get the package mode. At this blue package mode, you can add linear algebra. Package. So we need some of those function in there. So I just download existing package for linear algebra by adding this. And then once you finish, you just type backspace and you come back to the regular mode. So price squared bracket into the package download model to add that package, then backspace and come back. Okay, so that's the key. So you type this, you press square bracket, and then you have package management environment. And in this environment, you can start to add the package you want to use. Then you press backspace to come back to normal. Now you can using so after I call this package, the eigen function should work. So now I get my V and L out of the D using eigen function. So I have so eigenvalue is in first. Okay. So I should put a V next. Okay, so that's just introduction of Julia, and you're ready to play with this and maybe try some of this function multiplication between matrix. Any questions? So I type PWD with a bracket around it. So I call that a function. The function tells me what my current working directory is. So I know I'm under the folder of user JC CI. So I know my working folder right now is here. So if I have some files, I'm going to save to that folder so I can read that in. Okay. So next, we are going to download a Julia-based program and see how can we run it. So this program, I have to Google. It's a program published in Sales Systems in this paper by Talia Chang. And then what I will do is find the GitHub link, which is here, of the package they released. So I put this link to the chat so for you to visit. This program is developed under Julia language, and we need to first install a couple of packages in order to use it. So that's the way they suggest to. So we need this first, information measures. So I'm going to switch to my package management environment and add this first package. this package is developed upon other packages. So at the same time, the other package also installed. Okay, after this, I'm installing the second one. Do you have the same problem as start here on my computer?
Yeah, it's been sitting here for a couple minutes.
Okay, but it still went through on your computer?
Not yet.
Not yet. So if you don't use this package management environment, you can use, for example, using PKG. PKG. Remember, it's a case sensitive all the time. Then you can use PKG to add. So I already have the light. This will do the same thing, but you don't have to go into the package management environment. once I finish, the last package you installed will be the network inference. That's the package they developed. Okay, on my computer, I have already finished the installation of all these packages they suggest to install. So I think I'm ready to run. Again, I want to check my current working folder so I know where the file I'm going to read into from. Then from the simulated data set, I'm going to download a simulated data. So 100, maybe just this. Download. So I save this file under the folder which I can find. Okay. Okay, so what I did is I went to this subfolder called simulated datasets and found one. I think any of this should work. I just found this 100 E. coli 2 medium txt. I click this file and then click row. That's a raw file, and then I just save this as to the folder that I can find later. Let me know once you are ready, and then we can move to this notebooks. And there's info network. Okay, so once you have your data, we can come into notebooks. there's notebooks called info_network. So that's the source code we are going to copy and paste into this window here. So I can type that in using net tab network inference using light using graph. I can type number of gene 50, so assign a value to 50. So next, data set name is a string. So now I'm going to give the name I just download from the 100 equalizer to check the name of the file. txt. So it depends on which data set you download, and you may have a different name here. But remember, we need this extension, which is txt. So that's a full name of the file. We are ready to get in. Maybe this, I should place this as 100. I don't know. Let's see what's next. Then they use algorithm as a variable name, then the PIDC network inference. Remember to have this bracket around it. So that's the name of the function. They want to use this function pointer as algorithm, and then threshold. So next, they want to get genes. So this data set name has to be, well, get this. Data set name has to be in the folder that will be accessible for the program. So you can have this. I have trouble to paste this in there, so I still have to type. Mm-hmm. You have trouble to read that files in. Let's see. So your current folder is have to be under your name. Okay. Still trying. So you have your. Yeah, you saved here. Yeah, so this file you have to copy this and copy this. Yeah. And this PC, and the user, the C in C drive. Users, found your name and put it here. Yeah, I think then this should be able to find it. Yeah. Yeah, we just need to find the folder. Yeah, so this place we need a double quote. Yeah. No single quote in. Yeah. Yeah. Good. I'll try next, see whether I don't know whether my. Okay, good. It's doing its job. Okay, so if you put this called micro comment in front, that will timer the procedure. So that's inference. Oh, inferred network. Reason and genes. Yeah, so that's the way you just try and play with this. We're not going to go deeper, but I just want you to know how to do it. That's, I hope they can draw something here. Yeah, so I have, at the end, I have trouble to plot out this graph, and I don't know how to fix this. But this graph has already been inferred. We just don't have a way to look at it yet. Okay. Because some of these comparability issues, I don't know whether because of some package has a broken function name. So we cannot plot this graph at the end. But the graph, the network has already been created using the algorithm. Okay. Okay, that's all I want to share today in the clear language. I want to show you some of the examples how this Julia code looks like. So there are a lot of discussions about the side-by-side comparison between languages. Okay, if you decide to go further, Julia definitely a good computer language. I highly recommend it. Okay, I think that's all for today, right? Any other comments, feedback? Okay, you are ready for spring break, I believe. Okay, have a good one, everyone, and see you after spring break.